Welcome. Hello, everybody. We have several folks already. We will start the Kids First webinar. Welcome to the Kids First Winter webinar celebrating champions of discovery. The Gabriella Miller Kids First research program was inspired by Gabriella Miller, a childhood brain cancer patient and advocate. It was authorized by Congress in 2014 and funded with $12.6 million per year since 2015. The primary goal of the program is to build a cloud-based genomic data resource to accelerate collaborative research, leading to better prevention, diagnosis, and treatments for patients and families with pediatric cancer and structural birth defects. Birth defects and cancer are top causes of death in children. Studying these conditions together makes sense because there are common genes and pathways and higher cancer risk in children with birth defects. I'm Marcia Fournier, Kids First Acting Working Group Coordinator and Program Manager, and I will be your master of ceremony today. I will ask to put the slides on. I will go over the agenda quickly. Thank you. So agenda today, we'll start with a quick introduction about Kids First, followed by a panel of research experts highlighting examples of primary and secondary use of the Kids First data. Thank you, Dr. Adam, Azeez, and Olena for being here today. And this will follow with a demo on the Kids First portal featuring the Variant Explorer tool by Dr. David Higgins. Kids First is champion discoveries, and I will be telling you how. By supporting multimodal data generation, developing data resources, and supporting expert-driven research to increase the value of the data. Kids First Data Resource Center is developing data harmonization, data sharing, and cloud resources for data analysis. Kids First Data Resource Center allows for multiple data types and cross-disease associations. This is important because multiple data types allow a better view of the conditions underlying biology. Knowledge of common biology across pediatric conditions can speed up development of diagnostics, treatments, and prevention tools. Kids First commitment to sharing genomic data associated with clinical data allows genetic variant search and variant prioritization. This can be done within a single disease area or data set, across diseases and cohorts, allow users to explore similar underlying causes of cancer, structural birth defects, and other rare diseases. Expert-driven research grows a research community around Kids First data, which has shown to enhance data sharing and data usage and promote findable, accessible, interoperable, reusable data goals. Slide 4, please. Now I'm going to go over a little bit of the data cycle, where the data comes from. This is how sausage is made. Kids First invites new cohorts, and these cohorts are initiated by investigators like you for data generation with an NIH mechanism called EXO-1, which provides in-kind support to selected studies. Kids First prioritize cohorts that allows broad data sharing and brings rich information in phenotypes and clinical information. Since 2016, 79 patient cohorts have been selected for sequencing at Kids First sequencing centers, the Broad Institute and Hudson Alpha. While the program was initially focused on whole genome sequencing data, Kids First today supports the incorporation of additional data types, including RNA-seq, long-read sequencing, proteomics, methylation profiling. The majority of Kids First program spending has been on data generation. Kids First dollars also support the Data Resource Center and small research grants focus on improving Kids First data value. And I will talk more about that in a little bit. Kids First Data Resource Center plays a critical role on data sharing. The Data Resource Center compiles the genomes with phenotypes and perform quality check for NIH approval to deliver the data to investigators. The investigators have 6 months exclusivity with the data before the data is released publicly through dbGaP and the Kids First portal. Now, at this point, Kids First data serves as a basis for new hypothesis creation and new research grant applications. Overall, these efforts depending on collaboration. And I want to emphasize this aspect of the program, the data cycle of the program. It all depends on really harmonic collaboration between investigators, the sequencing centers, the NIH, and the Data Resource Center. And these collaborations, in turn, have, one, supported large-scale data generation from existing pediatric patient and family samples. Two, promoted secondary data analysis to enhance the value of the primary Kids First data sets. And three, led to the development of new and advanced analytical tools. Slide 5, please. To date, 37 studies, 25 structural birth defects, and 12 childhood cancer, including over 27,000 genomes from patients and families, have been released publicly via dbGaP and the Kids First portal. In this slide, you see a body graphic with conditions where Kids First data is available. Nearly 1,000 requests have been approved for secondary use of the Kids First data. These valuable data sets are catalyzing pediatric research and discovery. Harmonized data is available via Cavatica. Slide 6, please. Internationally, and I want to emphasize the international aspect of this program, both to submit applications for new cohorts data generation, but also for secondary use of the data. This is available to the international research community. And most of the users are in the United States, but we also have a significant number of users in India, Canada, Australia, United Kingdom, Germany, and Netherlands. Kids First is championing discoveries by driving other NIH grant awards. Over 10 NIH institutes, and this is slide 7, have made additional investments, issuing over 40 million in awards using Kids First data. The scientific work proposed by these awards are ongoing, and to date, Kids First program support was acknowledged in over 147 publications. As shown in this slide, the graphic, the first bar, shows that 92% of the funding for applications using Kids First data went to research project grants, and the great majority of those grants were R01s. So I'm going to tell you now a little bit about the grant opportunities. Slide 8. There are at least 2 research grant opportunities currently open to the Kids First community. The first one is a R03 that supports small research grants for analysis of Kids First data. This opportunity aims to engage the community in enhancing Kids First data value. The goal is to increase data value through development of new tools, analysis, and data enhancement. Slide 9. The second opportunity is a R01 research grant that supports functional validation of genomic variants including but not limited to variants identified to the Kids First program. And the receipt date for both the RO3 and RO1 are in February 2025. So please take a look at these opportunities and see if it's right for you. And we will have a link to the opportunities in the chat box. Now, I'm going to tell you a little bit how to access the Kids First data. There are multiple ways of accessing the Kids First data, as is shown in the slide. Kids First is championing discoveries by making the data more accessible. And we heard the feedback of the research community, and we now release a new landing page. And these improvements allow interaction with the data without login requirements. So I invite you all today to visit our Kids First new landing page and explore the data. The data is now explorable by non-bioinformaticians with summator level data available. If you are curious and interested in learning more, then you can sign up to the portal. And this first sign up only needs a valid email and about 15 minutes of your time. I've done myself. And you're going to have access to several analytical tools, including the variant browser that David Higgins will talk today. If your research, slide 11, please. If your research requires individual level data, additional steps are involved in assess those data sets. And the access is given through dbGaP. The dbGaP access approval of Kids First data is typically 2 to 3 business days, and this is possible thanks to a dedicated Kids First data access committee. So again, I invite you to explore the data and in multiple different levels. Slide 12, Kids First ecosystem is piloting cloud computing, including establishing cloud-based analysis space equipped with analysis tools for primary and secondary data users to facilitate the use of data. This effort was coupled with an innovative cloud credit pilot program to reach a wider user community to promote exploration and use of the cloud analysis platform Kavatika. So far, we have supported 22 cloud credit requests, multiple types of projects, structural birth defects, childhood cancers, and cross-condition projects. The average duration of projects is 3 to 12 months. And I invite you to learn how to apply. Just send us an email. This is a really quick 1 to 2 weeks approval process for cloud credits. And you can then go in Kavatika and test it out some of the tools available. More, slide 13, please. More investigation requested data in fiscal year 24 than any prior year. So the Kids First data secondary use is growing. Just as an example, from the end of September, which is the NIH fiscal year, to now November 2024, this number has already grew from 893 to 948 data access requests approved. Our quarterly reports have shown that most data sets are being continuously accessed. For example, in Q4, the fourth quarter of the fiscal year, approximately 80% of the studies received access request approvals, showing the utility of the data across structural birth defects and cancer data sets, as well as recent released data sets, but even data sets released prior to 2019 are still being used to date at quarterly basis with new request approvals. Slide 14. So far, Kids First data has yielded a number of research articles. That includes primary, secondary analysis, validation studies in model systems, and clinical research. So again, I'm here to invite you to explore all the resources that we are really excited to make available to the research community. This is another recent improvement in our web page. I invite you to explore the publication page and see examples of research being using the Kids First data. I also like to invite you to check the studies that are championing discoveries. These studies in slide 15 are DB-GAP studies most cited in publications, and both structural birth defect studies, including congenital heart defects, orofacial cleft birth defects, as well as pediatric cancer data sets like neuroblastoma and T-cell ALL. They have been accessed, and these are examples, again, of data sets. So I invite you to check these data sets, but also check all other data Kids First made available publicly through the Kids First portal. The Kids First is participating in slide 16, please, in promoting data interoperability, contributing to collaborative efforts across NIH. For example, collaborations with Include program, the NIH Cloud Platform Interoperability Effort, Common Fund Data Ecosystem, among others. The Kids First superpower is to bring to the table a data resource dedicated to pediatric data across pediatric conditions, putting kids first. Kids First is a trans-NIH program, and the working group is collaborating to championing discoveries in pediatric research. The working group is composed by NIH staff members representing multiple NIH institutes, as, for example, NICHD, NCI, and others participating. Special thanks to Kids First working group members for their dedication and hard work. And the bolded names individuals contributed to the portfolio analysis presented today. Check it out, the full list of Kids First working group members at the Common Fund website. Next slide, 18. I will conclude with the program vision, emphasizing the collaborative research and supporting data sharing. The Kids First hosted an event in collaboration with the Include program in a recent conference just last month. And this was a great example of the vibrancy and diversity of our community. The NIH is working together with researchers and advocates to bring discovery to patients and families across pediatric conditions. And I want to acknowledge the participation of patients and families in this recent event. All Kids First efforts are driven by constant program evaluation, informing continued improvement and innovation. So your feedback matters. Today, we're going to ask your feedback about this webinar, and it does matter for us. This will help us to shape the next events and next improvements on our portal and web page and data resources. I invite you to ask questions in the chat box. We will start answering them, but we will have a dedicated questions and answer after the panel. And actually, I want to ask you to add your questions to the Q&A box. So you can start. We're hoping that you're going to engage and have many questions for us. And right after the panel, we will address as many questions as possible. So at this point, I will thank you for your participation today, and ask Adam Resnick from the Children's Hospital of Philadelphia and principal investigator of the Data Resource Center to join me and introduce the expert researchers panel. Hi, Adam.
Hello, thanks so much, Marcia, for the great introduction. And thank you, everyone, for joining today. I had the privilege of being the moderator for today's champions of discovery session. In my mind, these are also going to be champions of collaboration. And we'll start off with Dr. Azeez Butali, who is the Gilbert Lilly Endowed Professor of Diagnostic Sciences at the College of Dentistry at the University of Iowa. He's also the director of the African Craniofacial Anomalies Network, a member of the Smile Train Research and Innovation Advisory Council. As I mentioned, I really think he represents, the preeminent example of the need for and the impact of global collaborations and partnerships. And as Marcia highlighted, you saw that the craniofacial cohorts within Kids First are some of the most highly cited cohorts within the platform. And with that, I'll turn over to Azeez to provide additional introduction, and then we'll follow up with Olena.
Good morning, Adam. Good morning, Marcia, Olena.
You're a little bit soft, Azeez. You have to just.
Can you guys hear me now?
There we go. Perfect.
Yeah. Good morning, everybody. Do I have permission to share? I have a few slides to share.
You should be able to.
Can you guys see my slides?
We can. They're great.
Okay, great. All right. Good morning. Thanks, Adam, for that wonderful introduction, and thanks, Marcia, for the overview of Kid First. And to Vanessa, thank you for the back-end work to get us to this stage. So today, what I would like to do is to share a few slides that gives a big picture of the work we do or we're doing using the genomes that we got from Kid First and additional studies. So the title of my talk is African Cleft Genomes, excuse me, from discovery to implementation. That means we are taking our data from the bench, we are taking them to the patients in the clinic and in the community. So can you see the second slide? Yep, good. So just a brief overview. In 2017, we were fortunate to have been funded by the Kid First, where we had genomes of 130 cleft triads from Africa. And in 2022, we got additional 860 individuals with whole genome sequencing data from the NIDCR. And using the data, we're able to identify de novo variants, which means variants that are not present in either of the parent, but in the affected child. And these variants were in genes that were highly enriched in processes that were relevant for craniofacial development. On the panel on the left here is processes that are relevant, like neural crest cell migration and cleft palates. And this data we had allowed us to be able to make that discovery. And more importantly, we're able to show that the genes that these de novo variants were found in are highly expressed in structures that are relevant for craniofacial development at different time points. So the first data I shared with you was what we found in terms of the coding region of the genome. We've also used the Kid First data to look at the non-coding region of the genome, where we've identified variants in the regions called enhancers, which are regulatory regions in the non-coding part of the genome that regulates gene expression. I'm just showing you, this is unpublished data, and this panel on the right here is the enhancer expression in the first buncher hatch in the HATS region as well, and this is in the wild type. But when we knocked in the human variant we found in an enhancer, it's abolished the function and the expression of the enhancer, suggesting that the human variant we found are functional. Again, this is the power of the discovery using the Kid First data set. And finally, as I close, I always ask the question in the clinic by the patients that what have you found in terms of what causes clefts in our kids? And we've always shared how far we've gone, and we've reminded our patients that discovery is the foundation for translation. However, since we have whole genome sequence data, which is 6 billion nucleotide for every family that are involved in our study, we also know that you can identify actionable secondary findings. So what we've been able to do is to identify, we looked at our cohort, and we're able to report that 2.3% of the variants in our cohort are actionable. And more importantly, recently we found that a child had hereditary breast cancer, a variant in a gene that increases the risk for hereditary breast cancer. and that variant was inherited from the mother. This is actionable, so we can now begin to pay attention to see if the phenotype, or to advise the family on lifestyle, and also bring them to the clinic to be able to educate them on what may likely happen, or to help reduce the risk. So again, this data allow that. And finally, like I said, the families that we work with are concerned about what's going on in terms of the discovery and what is actually going on now that they already have the kids born with clefts. And one of the studies that came out of our, genetic study was to look at their lives outside of the clinic. And in the study that we carried out, which is called the Photo Voice, which is the community-based participatory research, patients took pictures and documentaries of events that are going on outside of their life so that they can inform us on how best we can help them. And this is just to show you that our patients are still going through discrimination, isolation, because they have kids with birth defects. So that tells us that we have a lot to do in terms of advocacy. So I'll leave you with this QR code. For if you have time, you can scan this. This is a 15 minutes documentary of the patient experiences that we documented in our last project. And thank you so much for this opportunity, Adam and Marcia. I'll be happy to take questions. Thank you.
That was a great introduction. Thanks so much, Azeez. Let me turn it over to for a brief introduction to Olena. I've known Olena for a long time now, and she's really been a champion of data platform development and collaborative efforts. She is the co-founder of the Treehouse Childhood Cancer Initiative at the University of California at Santa Cruz Genomics Institute, where she is an associate professor of molecular cell and developmental biology. And really her focus has been on developing novel RNA-seq analytic approaches and tools that support clinical translation and additional discovery. I'll turn over to you, Olena, to further introduce your work.
Thank you so much, Adam, for that introduction, and thank you so much for having me here. I'm so honored to talk about Kids First data, because these data are so important and really enabled what we are doing, which is what I'm going to tell you about. So Adam mentioned the Treehouse Childhood Cancer Initiative that I lead at UC Santa Cruz. And so this initiative is a large data initiative. And so as Adam mentioned, we focused on RNA sequencing data across pediatric cancers. So we focus on large data sets. And so so here is my overview slide. And so the problem with studying RNA expression, and one rationale for studying RNA expression in pediatric cancers is that these cancers don't often have a lot of DNA changes. A lot of the changes happen in the gene regulation space, and so studying RNA measurements is very important. But RNA expression is relative, right? So you need to always compare different RNA measurements to say what's overexpressed, what's underexpressed, and so you need more data. And so the Kids First data have been crucial for us. We have integrated these data into our Treehouse compendia we made, and I'm going to show you a slide of that, of those compendia. And then we've also developed a pipeline we call Treehouse Comparative Analysis of RNA Expression, or CARE pipeline. And so this pipeline can take data from individual patients, so take RNA sequencing data from individual patients with cancer. And again, we work with pediatric cancers, and then compare these data to our compendia to identify targets for treatment, and to also help refine diagnoses for cases where diagnosis is not clear. So this cartoon illustrates the importance of Kids First cohorts, and importance of having large cohorts in general. And this idea, RNA expression measurements are relative, right? So if I show you a picture of just this one dog, you don't know, and I ask you, how big is this dog? You have no idea how big he is. I mean, unless you've seen the cartoon. But really, you know that this is a big dog if you see him next to some other dogs that are very small, right? And so now it's obvious that this is a really big dog, in fact, unusually big dog. And so we work with RNA expression data, and having large compendia of RNA expression data from other tumors is really helpful for us to find these big red dogs in cancer. So we often look for overexpressed genes in cancers that represent targets for therapy or could represent some important biomarkers. And this is how we spot them when we compare to other tumor types and see that this is now unusually highly expressed. So here is just a diagram of our compendia using our tumor map visualization. And so each dot here represents an RNA sequencing data set from a tumor. And so this includes a lot of data from Kids First, a lot of specifically pediatric brain tumor data that Kids First has. And so we've now integrated these data into our compendia and make them available to the community. We're also now part of the CCDI, so Childhood Cancer Data Initiative, which is an NCI initiative to create a data ecosystem that will connect all of the pediatric cancer data. And so Kids First Data Resource Center is part of this ecosystem, and we are as well. So now these data are shared in one environment to make even larger cohorts. Okay, so we've made these large cohorts, but I wanted to speak a little bit about the clinical utility. And so I've mentioned the SCARE pipeline we developed, which compares a single patient to these compendia. And so we have several case reports of clinical utility of this approach for patients. And I just wanted to share one case with you that really highlights this utility. So this is a rare pediatric cancer called myoepithelial carcinoma, has this particular biomarker. And so this is this particular patient was 23 months old, diagnosed with this cancer at Stanford, and so was treated with chemotherapy, delayed complete resection, but then unfortunately developed metastasis 3 years after chemotherapy. And because this is so rare, there is no standard of care kind of in that situation post first-line chemo, and not even clinical trials for this disease. And so we've analyzed RNA sequencing data from this patient using CARE, and so comparing this patient's data to our compendia, which includes Kids First data, and identified this candidate cancer driver pathway. And so here is some gene names here and some pathways. And so we postulate that this is a driving pathway in this tumor that contributes to the growth of this tumor. And so genes here in orange represent druggable targets. So, and you can see there is pazopanib can target these receptor tyrosine kinases, and ribociclib can target the cell cycle overexpression. And so in this particular patient, treatment with ribociclib, so one of these drugs, contributed to sustained remission, so produced a really dramatic clinical response where the patient was on this treatment for over a year, controlled the disease, and then he had surgery to remove all the nodules, and then continued ribociclib maintenance for another year, remained no evidence of disease, and now discontinued treatment and remains disease-free for multiple years. And so we're very excited about this case and other cases we have that are similar, really illustrating the power of having these shared data that Kids First is really enabling to help identify these treatment options for patients. And so I'm just going to end with my acknowledgements, our group and our clinical collaborators whom I mentioned, and especially Sherry Spunt, who was the treating physician for this case that I mentioned to you. So thank you so much for allowing me to speak today and to share about the importance of Kids First data, because as you can see, it's truly important. Thank you.
Thanks so much, Olena. You and Azeez, I think, did a fantastic job, highlighting a couple of things that I'd like to maybe reintroduce and maybe pull some threads, across both your use cases. So, despite really using two very different modalities of analysis, one for RNA-seq and the other for whole genomes, both of you really anchored into the utility of the Kids First, being that these large-scale data sets provide context for the analysis of the next patient or individual patients in that setting. Azeez, I wanted to start with you briefly, and maybe if you could just spend a few minutes talking about, one, the unmet need in the setting of craniofacial congenital defects, and why might it be important to have a global approach? So the Kids First program has now not only supported African cohorts, but Latin America, European ancestry, and Philippine-based cohorts. Can you talk a little bit more about, one, both the clinical unmet need, and then secondarily, how a global initiative across those cohorts is setting the stage for the next wave of discovery and care?
Yeah, thank you very much, Adam. So in terms of the clinical unmet needs, I think we've made a lot of progress around the world. Outside of developing countries, it used to be just surgery that is offered to children with cleft lip and palate. But now they are beginning to get comprehensive cleft care, which includes, orthodontics, speech, nutrition, and all that. And that's important because, as we say, a child that has been born should be taken care of while we're looking for prevention strategies for the kids that are unborn. So there must be a balance. So in terms of the unmet need, there's significant progress. If you look back 40 years ago, well, what was offered. So I must give kudos to all those in the field that have made this happen. I'm not going to mention names, but I know everybody has done so much around the world, including those that have gone to different countries and those that have taught people to fish by themselves, allowing local surgeons and local cleft team to do the work and collaborate with people across the Atlantic. Now, for collaboration, it's important to have multi-ethnic groups to accelerate discovery. We know that rare variants are population specific, and we know that there are differences in manual allele frequency. If you focus on just one population, you might be missing something, especially if that variant is not, or if the allele frequency of a particular SNP is very low in that population, depending on the strategies you're using for your analysis. If you're using GWAS, you're going to be looking at manual allele frequency. If you're looking at just rare variants are population specific. So either way you want to do it, you need more than one population. You need several populations well represented to be able to accelerate discovery. And that has been very helpful for our cleft work. We've discovered a lot of genes using samples from multiple populations. And those have been published. Like you highlighted, that we have a lot of citations for the Kids First Cleft program is because of the multi-ethnic cohort that has given it a lot of power and opportunity for discovery. So that would be my immediate answers to your questions. Thank you.
No, that's amazing. And, I think David will show this, but I think there are more than 95 million variants, across orificeal cleft cohorts within the Kids First platform. I was really intrigued, by some of your recent work and what you highlighted in your narrative, which is, the additional intersecting impact of genomic analysis, and you highlighted cancer predisposition, as a framework in that context. Can you talk a little bit more about that setting and, what is changing in this field for you and others, where you begin a path of diagnosis and analysis on behalf of a craniofacial phenotype, but then bring into that mix, a cancer predisposition or the cancer phenotype setting?
Thank you, Adam. So, like I said, the access to 6 billion nucleotide as a result of whole genome sequencing gives us a lot of opportunities to look at variants that are outside of the ones that contribute to clefts. So these opportunity for identifying secondary findings, looking at the genes that have been prioritized by the American College of Medical Genetics as actionable genes. For us, we took advantage of that and we asked those questions. And it was amazing that we found variants in genes that are predisposing for cancer. But also, we also found variants for other conditions. But that the fact that these are actionable, it means we can do something about them and we can, affect the lives of these families. Don't forget that cleft itself, in some regions of the world, is associated with stigma. Now, if you bring cancer on top of that, you're going to amplify stigma. It may not be a genetic issue, but in terms of marriage, a family that has risk for cancer, what happens to the daughters if they're going to get married? This becomes an issue beyond just what we're looking at variant. This is now a life-changing issue. So for us, it's comprehensive. We're not just looking at the discovery, we're looking at the lifelong effect of our discovery and relationship with the families.
Yeah, thanks so much for highlighting that. That really, this narrative that's within Kids First is not solely focused on diagnosis or causative variant identification, but actually has a potential for changing lifelong health and approaches for care. I noticed beyond the already defined cancer predisposition variants that in some of your recent publication, there is gene lists that includes genes that cancer researchers might care about. So beyond just already defined predisposition syndrome, like the BRCA gene, in one of your more recent publication, I noticed KMT2A, for example, as an implicated gene, which, if you looked at Olena's database, you'll find leukemias potentially are also altered in that context. Can you talk just a little bit about how you're thinking about the intersection between cancer and birth defects broadly as you start thinking about interpreting these new variants? And then I'll bring back to Olena. And I really want to contextualize this really in the broader mission of Kids First that really tries to harness a developmental context with potential shared ideology between them.
Yeah, thank you. So there is strong epi data coming out of the Scandinavian suggesting that there's increased risk of cancer in families with cleft lip and palate. That's epi. Now we have access to molecular data, to the genomic data. This is an opportunity to provide that evidence if we have enough data sets, and the intersection between these two data sets can help us really hone in on those risk variants that could increase the risk for both cleft lip and palate and cancer at the same time. And maybe that will be able to explain that epi data that was published a few years ago. So again, the Kids First is an opportunity. It's like a hub for us to be able to answer those questions and to be able to highlight them and at the same time find solutions. Because again, if you find the risk early and the phenotype is not present, for cleft, the phenotype is there because it's congenital. For cancer, it could be down the road, right? And you have the variant, and you know for sure this variant is going to lead to cancer. You can do something about it. They can begin to get some additional clinical observations, some tests. So that can mitigate it. So the discovery is actually accelerating prevention of the phenotype, which is cancer, even though the cleft is already present. So there's an opportunity for doing both and then for helping to reduce the impact of one while finding the solution for the other.
Yeah, and I suspect that just like you highlighted earlier about the distribution of, for example, craniofacial variants and their differences across global cohorts. Likewise, you might find differences in the associated cancer risk stratification and workflows there. Olena, one of the things that is really powerful about what you presented is that as much as the Kids First platform and other initiatives, are defining the genomic context of disease, that the interventional opportunities simply based on genomics can sometimes be limited. And significantly, I think, right, what you often are presenting is that in addition to the genomic mutations, integrating an additional mode of data layer like transcriptomics or proteomics can newly open a therapeutic intervention opportunity, for those patients. But I really loved your analogy to a big red dog. Can you talk a little bit more about some of your efforts and what it actually takes to drive that integrative analysis? So you mentioned your care pipeline workflow, but in order for someone like you to take Kids First data from Kids First and bring them together with CCDI data or other context, can you just walk us, the audience through what it takes, and then what might be ways that some of the new initiatives like you described, focusing on this API connectivity framework, might accelerate that process?
Yeah, sure. So we and you're right, Adam. It's you know, RNA-seq is like, we don't often think of it as pointing at interventions because I think it's tricky. So what we're doing is a tricky problem. So, you know, I showed the big red dog, but I think the biggest question that we've been struggling is what are the right dogs to compare, right? And so in the context of these cohorts, what are the right samples to compare each patient to identify clinically relevant overexpression? Because, you know, you can compare something to something and always find overexpression, but what is the right? So maybe that's our driving question that keeps us up at night, is what's the right cohort for comparison? Is it all cancers? Is it normal tissues? Is it a selection of similar cancers? And if you do all of that, you get different results. So this is something and I don't have an answer right now, but this is an active area of investigation for our group. What would help with this, and that where CCDI, I think, is so important, is we need as large of a cohort as possible. You know, the larger, the better, just because there are so many variables. You've been talking with Dr. Butali about ethnicity, right? And we know that gene expression, I mean, there's so much variation across different ethnicities, and we're currently not capturing all of that variation in what we have. So we definitely need more data from diverse populations, from diverse diseases. You know, really my dream is to create a universal RNA-seq map of all the data. And, you know, for RNA-seq in particular, you know, what I'm not mentioning is the data needs to be processed using the same pipeline, right? So for it to be comparable. And so Treehouse and Kids First actually have very similar pipelines. But then, you know, some other places may have a different pipeline. And so what we do in Treehouse when we collect data from multiple sources is we reprocess it with our standardized pipeline so that it's comparable apples to apples. And we also do QC on it. So we do quality control. We have defined specific depths of sequencing we need for this type of analysis I presented. And so we run a rigorous control before we include a data set into our compendia. And so that's very important. So I think we need more data. We need to standardize the data. So we need to, as a community, maybe decide what's the right pipeline, or when do we really need to recompute, because recomputing with another pipeline, of course, costs resources. And if you have a large data set, that's going to cost more. Sorry, am I answering your question? Is that
Oh,
what?
no, it's perfect. Yeah,
Okay.
no, I really definitely agree. And, you know, I think part of the reason I asked you that question is because I think, you know, what Treehouse really represents is a model of this emerging ecosystem that Kids First is part of, which is, you know, as much multimodal data that the Kids First data resource provides within its own platforms. I think the program is actually very advocate of collaborations and interoperability in ways that Treehouse is an exemplar, meaning, you know, having a team like yours deeply think about, strategize, and support RNA-seq as a data modality for intervention that leverages the Kids First data resource but contextualize it on behalf of a therapeutic approach is, you know, phenomenal. I think similarly, you know, Azeez, like in your work, you know, obviously you're leveraging Kids First whole genome sequencing data, but again, the craniofacial community has FaceBase and other platforms as well that, you know, you again interact with and collaborate with in the context of Kids First. So for me, both of you sort of represent this broader ecosystem of collaborations and platforms that advance either disease-specific or data modality-specific initiatives, harnessing the multimodal and cross-disease landscape, you know, within Kids First. I'll give both of you a chance briefly to you know, you've both been leveraging Kids First data, and this is an open question. I'm not sure it was one we actually prepped you for, but you know, what would you want to happen next? I think, Olena, you maybe hinted at it, but I'll give you a start from there. I think you mentioned more data. What else?
I would, you know, say more clinical annotation as much as possible. And I know some of it is tricky to share, right, because of consent and because of identification. But, you know, genomic data is nothing without rich clinical annotation. I think we've all understood that. So I think as much as possible, if we can collect clinical annotation and follow-up data, I think that would be super useful, and then we can make more sense of the genomic data. So that's one thing I would really, if I had one.
And I think that's great. I think that's one of the key areas of focus for us programmatically is to recognize that genomic data is point-in-time data, but that really clinical data is longitudinal in nature, and really developing and informing on that longitudinal continual data integration as paired with genomic is really the next wave of opportunity for advancement of translation. Azeez, I'll give you a chance to set the stage for the future.
Well, I think there's opportunity to at least get additional ethnic, multi-ethnic data. I don't think we've covered everything that I would love to. For example, my African data just has West African cohort in the whole genome sequencing, and Africa is very diverse, just as one continent. I'd love to have data set from East Africa, from South Africa, from Central Africa, from North Africa. But again, I know it's one day at a time, but by the time we have this comprehensive data set across the world, that's representative, I think that will help accelerate discovery. On the clinical side, I think Adam, your team is doing a good job trying to push us on this side to get you more clinical data so that we can, just oppose the two to be able to accelerate discovery and then improve lives. So I think that will be my proposition for the future. Thank you.
Thank you both. And so I think with that, let me see if there are any additional questions in the Q&A. I don't see any more questions that I missed, but Olena, Azeez, thank you so much for all the work you're doing, again, for your spirit of collaboration, engagement, and partnership, and really demonstrating the impact and power of the Kids First program. We're thrilled to have you as collaborators of the DRC, but even more so, thrilled for all the work you're doing on behalf of the participants and patients within the program.
Thank you so much.
Thank you.
It's an honor to
Thank
be
you.
here.
Yeah,
Thank you very much, Adam. And thanks, Olena. Nice to meet you here.
Nice to meet you. Thank you.
Bye.
Bye.
Thank you, Adam, Azeez, and Olena, for your participation. We will follow them. I will give a few minutes for questions.
Yeah, I checked. I think we covered all the questions. And it may be that
all the questions. Okay, great.
People could have additional question as David is presenting, and I'm happy to either answer them directly in the Q&A or we can bring them to open discussion.
Wonderful. And we will now I would like to invite Dr. David Higgins, also from the Children's Hospital of Philadelphia. So David, the podium is yours. We will have a demo on the portal covering our Variant Explorer tool.
Thank you so much, Marcia. Thanks for having me. It's so great to be here at the Kids First Winter webinar. As Marcia mentioned, I've got a couple slides to set up my demo, but I hope to spend most of my time today on the Kids First portal, showing some of the features that we have that can help identify and support research projects exploring different variants in Kids First datasets. So as Marcia mentioned, my name is David Higgins. I am a program manager, and I work at the Children's Hospital of Philadelphia in the Center for Data-Driven Discovery in Biomedicine. And so I am the person who's here, and I'm here today representing the Kids First Data Resource Center. But I am just the tippy tip of the iceberg, or the guy at the front door who's standing in front of a very large group of people. And so I'm so honored to acknowledge the contributions of all of our different teams, of all of our different sites who built together, who worked together to build the Kids First Data Resource. So there's a big list of many of us who work as part of the Kids First DRC. And so Marcia mentioned this earlier in her slides, is that more researchers are requesting Kids First data than ever before, which is exciting. So more people are using the data, more people are interested in the data sets of Kids First, and more people are incorporating it into their research projects. So at this point, we've had 266 requests approved so far in 2024, which is the most of a calendar year. And we're up to almost 900 all time. And I'm sure when we pull the updated numbers at the end of the calendar year in January, after all the final numbers are in, will be even higher than we are now. So more people are using Kids First data, which is very, very exciting. And so, of course, what we ultimately are hoping for is not just people are looking at the files, but people are using them and making discoveries and then publishing those discoveries as well. And so I'm also excited to share that, yes, people are publishing their data, are publishing findings on Kids First data as well. So this is our papers, individual calendar counts for each individual calendar year. And so 2023, we had a really great year. 2024, again, we're off to a very good start. Again, these are only our numbers through September, but then when the year closes at the end of this month, we pull the numbers again in January. I'm expecting that we'll be, again, perhaps the greatest year yet for research publications. So very, very exciting time to be working and using Kids First data. So I wanted to feature some of these recent articles just to kind of highlight what are the types of research that people are doing on Kids First data. And so I'm just going to share some titles here, but I can pull out a link here and get it into the chat so that way everybody can follow up and read these publications on their own. Here we go. And so, so obviously Kids First data are being used for genomic discoveries. And so this is what Adam and Azeez were talking about at the beginning. So here are two recent articles that came out in 2024. This big landscape article identifying T-cell acute lymphoblastic leukemia that came out this year, and on the cancer side, and then on the structural birth defect side, there is a study from the NeuralTube group. Identified risk of meningomyelocele is mediated by the common 22q11.2 deletion. So again, kind of this very broad, in order to be able to treat the condition, we need to understand the underlying genetic causes of them. And so here's two recent papers where we started that discovery process. But Kids First data sets are also, findings are being validated in model systems. And so a lot of these initial studies are just kind of scanning the surface and finding interesting sites to investigate further. And so at this point, it's exciting that researchers are not just, beyond the survey, now we're starting to dive into these most interesting locations. So similar to like the data that Azeez was showing, where we were looking at the, expression of these non-coding variants and how does that influence expression of genes. And so, again, here's two examples, one from a cancer study and looking at neuroblastoma. They found in ways that ALK upregulates other genes in the signaling pathway. And they did that in mouse models. And then in a human cell model, they did functional characterization of genes that are associated with human heart enhancers. And so they were able to, again, test expression of genes in this cell line model by making very specific mutations that mimic or identical to the patients that were found in patients from the heart defect study. So taking these survey findings and then testing them and validating them and doing more discovery. And then the last kind of broad set of articles that I'll talk about is that Kids First data sets are being used to explore the secondary genomic findings, which is again part of our panel discussion a little bit as well. So here's two studies that came out. The top one came out this year in 2024. It was based on a pediatric cancer study where they did secondary genetic findings. And then the second study, this is actually a study from Dr. Butali's group that came out last year in 2023, where they were looking at these clinically actionable secondary findings in families from sub-Saharan Africa. with non-syndromic orofacial clefts. So broad types of different research that are being used on Kids First data. And again, after the webinar today, I encourage you to check out the website and read more about all the exciting ways that people are using Kids First data sets as part of their research programs. So to dive a little deeper into these secondary genomic findings, and explain what that might be for folks on the call who aren't familiar with what those findings are. I have this kind of scenario here where a patient with a cleft lip might consent to join a study to identify the underlying genetic causes of orofacial clefts. And so as part of that, the patient's entire genome is sequenced and that data is delivered back to the researchers. And so the researchers have the mutations of the participant's entire genome, and they can survey it and identify variants in all genes. And so in addition to the genes related to craniofacial development, what the researchers identify is that this patient also has a mutation in the gene BRCA1. BRCA1, if you're familiar, BRCA1 is known to increase, or mutations in BRCA1 are known to increase the likelihood of developing breast and ovarian cancer. And so because this gene is known, and it's not the subject of the original study, which is looking at orofacial clefts, this mutation would be known as a secondary genetic finding because it's unrelated to their original diagnosis. And so depending on the experiment's protocol and depending on how the patient consented, that patient may be notified of the secondary genetic finding by the researchers carrying out the study. So as I kind of talked about, there's lots of, or two different papers that have come out in the last 2 years using Kids First data to look at these secondary genetic findings. And so in order to be able to do this type of study, how would you want to carry out this experiment? So the American College of Genetic Medicine publishes a list of genes like BRCA1 that have known associations that can be classified as secondary findings. And so if your research question was broadly, which of these genes have variants in Kids First cohorts and what's the relative frequency of these variants? But in order to be able to do that experiment or answer that research question, you would need to receive access to the Kids First data sets via dbGaP. Then you would want to identify harmonized VCF files for those individuals of interest on the Kids First portal. Then you would query for variants in that list of genes from the ACGM in order to be able to pull out just those variants. But then you would want to then annotate those variants further for deleterious effects through databases such as ClinVar and VEP. And so it would take many steps to kind of carry out all these different methods to be able to narrow down this list of genes to find potentially variants of interest. And so today, though, what I'm going to show is that we can actually do a lot of this very rapidly and quickly using our tool called the Variant Explorer on the Kids First portal. And so all through the Kids First portal, just by pointing and clicking and applying some filters, we're going to be able to identify genes or variants in genes from this ACGM list and be able to see what different studies those particular variants are present in. And so everything I showed today is you can do with me at the same time. You could log in later today and do it as well. So all you need is an account on the Kids First portal, which is free to sign up. There's no additional data access requests required for what I'm going to show today, though I'll explain at the end how applying for data access could unlock additional information at the end of the demo. And so with that, I'm going to end my slideshow for right now, and I'm going to navigate over to our website, the Kids First portal, portal.kidsfirstdrc.org. And so let me just shuffle some things around here on my windows. And so if you're interested in following along, there is our link to the Kids First portal. And so once you follow that link, it takes you to our portal website. We've got a great new cover page that introduces what we had, the types of data that we have inside the portal as far as which different studies, how many participants, things like that. We also have information about the types of conditions that patients are diagnosed with inside of our portal. So it's a great new page. Be sure to read through it. So today I already have an account, so I'm going to log in. If you don't have an account, it's very quick to sign up within minutes. And again, there is no cost to access the data on the Kids First portal. So whether you log in or create an account, it will ask you to use one of these 3 features. Anyone with a Google authenticated email address can create an account. If that's an institutional Google-based email or even at gmail.com email address, you can log in with that. If you have an ORC ID, you can also log in with that. And if you have an NIH ERA Commons ID, you can also use that to log in as well. So I'm going to log in today with my NIH RAS, and it's going to query my username and password. And then I'm going to accept the conditions. And when that completes, then it connects me into the Kids First portal. And so the Kids First portal has lots of different features to explore different studies and our data exploration tool. But today I'm really going to focus on the variants. And so the variants tab, or our variants explorer tool, allows you to kind of quickly survey and query variants in Kids First participants, all within the Kids First portal, without touching the command line, without opening a BCF file, just by kind of quickly doing a couple commands that we're going to carry out. So right now we can look and we can see that there's 411 million different variants across all of our different Kids First participants. So there's lots of variation in here. And that's a lot to start with. So we can try to narrow down, well, which one of these might be most interesting to us. And so that's what we're going to work on today. So our example is that, if we wanted to carry out a secondary genetic finding study similar to those recent publications that I shared, we could start by looking for variants in those genes of interest that are already labeled by the ACGM. So I'm going to bring in another window here into my demo space. And so what we can see from here is that, so this is a table that I got from the ACMG's recent publication where they updated their version 3.1 gene list. And so they have a list of genes that are associated with particular cancer types, a list of genes that are associated with particular cardiovascular phenotypes, and then different metabolism phenotypes, as well as different kind of this catch-all miscellaneous phenotypes category. And so what we can do is that we can go ahead and select all of these genes out of this table, and we can copy them. And then I'm going to navigate back to the Kids First portal, and I'm going to apply a filter where I just want to see the variants in those genes. So you can see this gene column right here is labeled. So I'm going to go ahead and click the gene column here, and then I can search by gene. So if you have a single gene that you're interested in, you can type in the name of your one gene. But we have a lot of genes today, so we're not going to do single. So we're going to upload a gene list. So I'm going to click that upload button, and then I'm going to paste that list of genes, and that gets me 86 genes in our portal is recognizing. So we're going to upload that and click the button. And so now you can see that if I close out my window there, that we've narrowed down our number of variants here. So we have 1.1 million variants in this list of 86 actionable secondary finding genes as published by the ACGM. So we can narrow this down further because many of these, so it's filtered by genes or variants that are predicted to be deleterious, but we can narrow this further. In fact, that's part of the processing that would be required to be done, is that we're interested in variants that have been annotated by ClinVar as being pathogenic or likely pathogenic. So we can do that as well. So we can add a second filter for pathogenicity, and then we can select ClinVar, and then we can apply 2 filters for variants that are either pathogenic or likely pathogenic, and click apply. And so this narrows us down. We can then to 914 variants. So we're getting more and more specific here as our query continues to narrow. And so I could scroll over a little bit and show our table that shows the different features of what we have. So here's our list of genes that we got from our published list. And then this is the consequences of these particular variants. And then out here, this is the annotation with ClinVar. So these are labeled as either pathogenic or likely pathogenic. And then if we scroll over to the right, this is the number of participants across all Kids First cohorts, and then the number of studies that include at least one affected participant. So many of these are very rare. They're only present in a single participant in a single study. But we could narrow this down further. So since we've been talking about orofacial clefting, we could apply an additional filter. So maybe we're just interested in other orofacial cleft studies on the Kids First portal. So we'll add one last filter here, where we might just be interested in variants from our orofacial cleft Filipino ancestry, our orofacial cleft Latin American ancestry study, and our third one, our orofacial cleft European ancestry study. So we could apply that, and now we're down to 100 variants. So these are variants where they're present in at least one participant in one of these 3 studies that's also been labeled by ClinVar as pathogenic or likely pathogenic. And so it allows you to then narrow down. Again, many of these are very rare, where the number of participants is very low, and the number of studies is also low as well. So we're interested in specific details. So for example, this variant here is a frame shift mutation in this gene GAA. We can learn more about it. So we can click on this variant, and it brings up the variant page where it tells us information about it. And so we can learn that this particular mutation is a protein coding mutation. It's associated with glycogen storage disease too, and then it links out to OMIM if you wanted to read more about that and learn what that is. It gives us the nomad frequency of this variant, tells us a relative frequency, and then it gives us information about which Kids First studies this variant is found in. So it tells us that 2 of the almost over 1,000 participants in the orofacial cleft European ancestry study have this variant. And then likewise, but it's also present in participants in other studies as well, including the diaphragmatic hernia study, the leukemia study, the intersections of cancer and birth defect study, and our brain tumor study as well. So it allows you to not just identify, you can find what we're querying, but then also see how it connects to other studies as well. And then it gives information about the frequency of this variant in other cohorts as well, including nomad and 1,000 genomes as well. So it kind of gives you a way to be able to quickly survey and explore genes very, very rapidly and see which variants are present in other Kids First studies on a quick method using the Kids First portal. So right now what we've been doing is kind of recreating and copying that list of genes that we have already been identified. So these are already known associations with these particular genes and these particular conditions. And so if we wanted to go more into discovery mode, you can also use the Variant Explorer to kickstart that as well. So I'm going to do a new query. If you click that new query button, it saves that first one. So that 100 that we identified, that's still right there up at the top. But then we're going to create a new one and kind of start from scratch. So maybe we're interested in the same 3 studies as well. So we can start by applying another filter for that study code. And we'll give it a second to load. And so we're going to be interested in the same 3 studies that we chose before. And so the orofacial cleft European ancestry, the Latin American ancestry, and the Filipino ancestry. And click apply. But then instead of using this published list, let's try to discover on our own. So maybe we're interested in variants that are known to be, or not known to be deleterious, but would be predicted to be deleterious as far as impacting the protein's function. And so we can use that by applying one of our pathogenicity filters. And so, for example, we can apply a filter for a CAD score. So CAD is a database that helps to kind of predict and quantify the effects of mutations on the function of a particular protein. So we can filter based on CAD score greater than 30 And what this would do would then basically, it's a fairly restrictive filter that would only narrow or identify variants that are kind of predicted to be deleterious, predicted where the protein would not be functional, and it would have some type of effect on the particular patient. And so that narrows us down very rapidly. Now we're at 16,000 participants. But if we wanted to apply further, you can see that some of these are fairly common. And so we could then, well, what are the nomad, the allele frequency of these, which is where we only want to look at rare variants. So we could apply a filter for that using the frequencies. We could apply a nomad filter, or we're only interested in variants that have an allele frequency of 0.0001. And click apply. And so that gets us down to 6,800 variants of interest that meet this criteria of we've identified, present at least one participant in these 3 studies has this variant, and that it's predicted to be deleterious based on CAD, and then it's relatively rare based on nomad. And so, of course, you can adjust all these settings based on your own search criteria as well. And so as you scroll through, we're kind of past this survey mode, and now we're in discovery mode. So a lot of these genes don't have annotations in ClinVar, though some of them do. And so it kind of gives you this opportunity to say, well, perhaps, what is this rare variant doing? How is it affecting the participants with these particular conditions? So I will click on one here in order to wrap up my demonstration. And so let's click on this particular variant here. And so this particular variant is in this gene called ELOVL3. It gives us information about what this gene is, and it tells us it's present in 4 individuals across 2 different projects or 2 different studies. The orofacial cleft European ancestry study, 2 participants have it. And then also participants in the neuroblastoma study have the same variant as well, 2 of them as well. It gives us some information about what different conditions are associated with this particular gene too. And so to wrap up, the Variant Explorer is our explorer tool. So it allows you to survey and explore very rapidly and get a sense of what types of variants are out there. So the limitation of the Variant Explorer, and this is where the demo ends, is that we're not able to directly identify which of those 2 participants have this particular mutation. We just know at this point that 2 of the 1,100 in the orofacial cleft European ancestry study have it. So at this point, you would then need to receive access to the data in order to be able to kind of directly connect the participant identity with the genomic information as well. And so in order to do that, you would want to go onto the studies page, and then you can look up those same codes that we've been using before. And if you are unsure of the codes, you can use the studies page to see that connection. But we would then find, the orofacial cleft European ancestry study, and then that links out to dbGaP, and that's where you would be able to apply, submit your own data use application, and then that would be how you would then kick off your own project. From there, you could then, after you receive access, you could then go back to the Kids First portal and search using data exploration tool to find participants from that particular study. So we could type in our study code, KF orofacial cleft European ancestry, find those participants, find the data files of interest. So then you could select the particular data files that correspond to, for example, the GVCFs, 1 per participant. And then from there, you would be able to analyze in Cavatica using Cavatica's tools. So that would be the one way to do it. And then the alternate way would be to use the Variant Workbench, which is our new tool that we've launched this year, which allows you to be able to continue to query similarly to these large tables that we have in Variant Explorer, but you can make additional connections to participants and their data as well. So the Variant Workbench would be another tool, which I don't have time to go into my demo today. So to wrap up, if I can have 60 more seconds just to conclude here. I would love to share with you all that if you have additional questions, we'd love for you to reach out and speak with us a little bit further. And so we have a monthly office hours session where us and our partners at Valsera meet on the third Thursday of every month at 2 o'clock Eastern time to talk about anything that you're interested in talking about. So if you have any questions, what is this variant workbench? I want to learn more. How do I recreate that same demo that you did? I'm thinking about getting started, but I don't know how. Anything that you have is a great time for office hours. So please stop by. They're on the third Thursday of every month at 2 o'clock Eastern. So our next meeting is going to be 2 weeks from today. I know that's kind of tight with the holidays and things like that. People are wrapping things up before the end of the calendar year, but we do it every month. So if December doesn't work, then please come back in January and talk to us then. And then finally, our support email address at the bottom is always available throughout the month, support@kidsfirstdrc.org. We've been trying to do a really good job this year of identifying and responding to folks through the support and tracking and making sure that requests are being acknowledged this year. So please reach out. If you don't have a chance to make it to office hours, shoot us an email and me and my team will be happy to reach out and speak with you. And so then with that, I'll conclude again by acknowledging all the folks who went into this demo that I shared today. Through their combined efforts that we have this Kids First data resource and hope that we can make great discoveries to help kids with all these conditions. And so with that, I'll pass it back to Marcia and allow her to wrap us up.
Thank you, David, for the amazing demo, and I'm getting better and better on keeping up with you. Hope everybody enjoyed and log in and explore the Kids First data. Do we have any other questions? I think I saw a raised hand, but we may already have responded the questions. So if we don't have any other questions, I'll give time back to everybody. I think nobody's going to complain about that. And then thank you again to our panel of speakers, Adam, Olena, Azeez, and David for being here. I learned so much and I'm looking forward to continue championing discoveries with you all. Thank you.
Thanks so much, everyone.
Thanks, Marcia. Thanks, everyone.
Bye.
Bye.